Welcome to Research Geek. Today I will brief about fluorescence AccQ-Tag method for amino acid analysis by reverse phase HPLC and determination of L-Citrulline, L-Ornithine and L-Arginine in the provided samples. First of all, standard curves for L-Citrulline, L-Ornithine, L-Arginine and the contents in the samples were determined by AccQ-Tag pre-column derivatization method, that is by RP-HPLC. AccQ-Tag reaction was performed at 55 degrees centigrade for 15 minutes. 4, 12, and 36 ppm of L-citrulline, L-ornithine, and L-arginine were mixed 1:1 with 4 ppm L-serine as internal standard, so that 2, 6, and 18 ppm each of L-citrulline, L-ornithine, L-arginine, and 2 ppm of L-serine was formed. Samples were filtered through 0.45 micrometer filter and diluted 5,000 times. Pre-column derivatization reaction. Standard sample 10 microliter, borate buffer 70 microliter, and it was vortexed for 10 seconds briefly, and then spin down. Then AccQ-Tag reagent was added, that is 20 microliter. It was again vortexed for 10 seconds, spin down, and incubated at 55 degrees centigrade for 15 minutes. Mobile phase A is the acetate phosphate buffer, 20 millimolar acetate and 6.25 millimolar hydrogen phosphate, pH 6.8. Mobile phase B, acetonitrile absolute. Post run 10-minute washing with acetonitrile followed by 20-minute equilibration with mobile phase A. Excitation 250 nanometer, emission 395 nanometer. Gain used was 100. Attenuation 4, filter 5 seconds. Injection volume was 10 microliter. What is AccQ-Tag chemistry? What it is? AccQ-Tag chemistry is a pre-column derivatization method developed by Waters Corporation. It is designed to make amino acids detectable by HPLC, UPLC with optical detection. Most amino acids lack strong chromophores. They do not absorb UV visible light well. Then derivatization adds a fluorescent tag so that they can be separated and quantified. How it works? Proteins or peptides are first hydrolyzed into free amino acids. Then these amino acids react with AccQ-Tag reagent, that is AQC. This reagent binds primary and secondary amines, and the reaction produces stable fluorescent derivatives. The derivatized amino acids are separated using a special AccQ-Tag column. Here we use C18 column, and fluorescence or UV detection is used to quantify each amino acids. Advantages: it has high sensitivity, detect amino acids at very low concentration, stability, derivatives are stable for extended periods, unlike some other methods. Reproducibility, standardized kits and protocols ensure consistent results. Versatility, used in biopharma, food testing, proteomics, and cell biology. It's very versatile technique. Run parameter, time percentage mobile phase A, percentage mobile phase B. So this is the run parameter I used. Flow was 1 ml per minute, flow rate, and column temperature was 35 degrees centigrade. These are the results. For L-arginine, x-axis and y-axis was determined as described in my previous briefs. So Ca is the concentration of analyte, like 0 ppm, 2 ppm, 6 ppm, 18 ppm. CIS is the concentration of internal standard, that is L-serine, so 2 ppm in each. AA is the area of analyte, that is analyte means L-arginine, and AIS is the area of HPLC, area of internal standard, that is L-serine. So for x-axis, concentration of analyte was divided by concentration of internal standard, means that is analyte 268 was divided by 2, so it comes to 0.139. And in y-axis, area of analyte means area of L-arginine was divided by HPLC area of L-arginine was divided by HPLC area of L-serine. So this after dividing HPLC area of L-arginine by HPLC area of L-serine, then that comes to these values. So this is the chart for L-arginine, ppm L-arginine 0268, area of L-arginine. These are the area of L-arginine. These are the area of L-serine, and x-axis 0.139, y-axis these are the values. And for calculation, these for x calculation, these values like 1.067850.277 was divided by slope of L-arginine by 0.6318. So it comes to 1.690171379. Then L-arginine ppm was determined by multiplying these values, x calculation by ppm of L-serine, that is 2 ppm, means 1.69 means 3.38. Multiplied by 2, it comes to 3.38. Then final L-arginine ppm, L-arginine final was this much, because it was 5,000 diluted times diluted, so it was multiplied by 5,000, and finally L-arginine in milligram per deciliter. Then ppm was changed to milligram per deciliter, means these values were divided by 10, and it was now again divided by 1,000 to obtain the concentration in gram per deciliter. So these are the values in gram per deciliter. Similarly, for L-ornithine, x-axis and y-axis were determined. Slope was 1.0802. So for L-ornithine, slope was 1.0802. And again, x-axis and y-axis were determined to plot the standard graph. So y-axis was plotted against the x-axis, and its chart is like this. L-ornithine ppm 0268, area of HPLC area of L-ornithine, these are the values. Area of L-serine, these are the values. The x-axis was obtained by dividing the ppm of L-ornithine by ppm of L-serine, that is internal standard, so it comes to 0.139, and y-axis was determined by dividing L-ornithine area by L-serine area. So these are the values, and then standard graph was plotted, x-axis and y-axis, then slope was obtained. So for x calculation, this y-axis value was divided by slope 1.0802. So then x value is obtained, and L-ornithine ppm value is obtained by multiplying x value by 2. That is the ppm of L-serine. Then final L-ornithine was obtained by multiplying this L-ornithine ppm value by 5,000, because it was diluted 5,000. Sample was diluted 5,000 times. And final L-ornithine in terms of milligram and gram per deciliter was obtained accordingly. Similarly, same process was repeated for L-Citrulline. Its slope was determined. It is 0.5597, and the values for different samples like sample 1.1, sample 2.1, sample 3.1, sample 4.1 values are obtained, and its readings are determined as described as before. So this is the L-Arginine, L-Citrulline, and L-Ornithine concentrations. Sample 1.1, sample 2.1, 3.1, 4.1. These are the values for L-Arginine, L-Citrulline, and L-Ornithine.